Welcome to another year of on-farm research. I'm glad to see all of you guys here. My name is Melissa Carlson, if we haven't met before, and I am the research coordinator for Minnesota Wheat Council and our on-farm research network. So this is our annual on-farm research summit, where we will go through all the data from this year and our trials that we did this year. So glad to have you guys here, and we'll be following along in the book, at least for the first half of this morning. So when I'm talking about all the on-farm data, so everything that is on the PowerPoint is also going to be in the books that you guys have in front of you, if you want to follow along, to make it a little easier to see some of the data. Before we get started, CU credits. I'm sure you guys saw, if you are looking for CU credits, I'm sure you noticed that the sign-in sheet is over by the front here, so make sure to scan that or sign it if you need to fill out on some extra credits. And then just in a quick note for later this afternoon, lunch, after we're all finished here, lunch will be up on the concourse. You will need a ticket for lunch, so make sure if you haven't stopped at the registration desk to get your name badge and your meal tickets, make sure you do so before you go up to lunch. Otherwise, you'll have to walk all the way back down here. All right, and with that, we shall get started. All right, so the On-Farm Research Network, I'm sure a good number of you in the room are familiar with what it is, but for those who are not, it is funded by the Wheat Checkoff. So the Wheat Checkoff is 2 cents for every bushel of wheat that gets sold. So we invest that into, the council invests that into research and wheat promotion overseas, in the nation and overseas, and so part of that checkoff funds also goes to fund our on-farm network, in addition to university research and wheat breeding. We are also grant funded, so we've had, several different grants over the years as we've done all of the different trials that we've done. So this year, some of the trials that we did were funded by SARE, which is the State Sustainable Agriculture and Research and Education Council, and they fund a lot of soil health, cover crop kind of research. And then we also had one project that was funded by a Minnesota Corn Innovation Grant. And in the past, we're very grateful. MDA and Minnesota Soybeans have also helped us out a lot with grants and working together on projects. And we cannot do this alone, so I definitely, right off the bat, want to give a big thank you to everybody who makes all of this happen. First and foremost, all of our trial cooperators, the growers who are putting these trials in. We try to make get everything prepared ahead of time and make it all go as seamlessly as possible out in the field so you guys don't have to spend a whole lot of extra time putzing with trials. But it does take a little bit of patience just to get out there and put some strips in and harvest them at the end of the year. So I definitely thank all of our growers for that. Our advisory committee members, so the On-Farm Network has an advisory committee of several growers, crop consultants, and a few industry folks, and University U of M Extension on there to kind of direct the trials, kind of finalize. We get ideas for trials from growers and people that we talk to, and then the advisory committee helps me to narrow down and refine what we're actually going to study and kind of finalize the protocols for the treatments that we're going to look at. Also, definitely want to thank Magnuson Research Farm, Gary Purath, and West Central Ag for borrowing us their weigh wagons every year, which are very essential to harvesting these trials. And then we work with Central Consultants, to especially Shane Weller, Ben Generex, and Ethan Holst are the three main guys that we work with. And they've been very, very helpful over the last however many, 5, 6, 7 years doing trials with them. So very grateful to them. The co-ops that help apply the fertilizer. I know it can be a little putsy for them in their busiest season, so I'm definitely grateful to all the co-ops. Red Lake County Co-op over in Red Lake Falls this year definitely helped us out a lot this year, so definitely appreciative to those guys too. And then Dufo Spray Co. and the Northwest Research Outreach Center and Rock Farm crew was also awesome this year in helping us do some kind of small plot, large plot collaborations. So it takes a huge team to get this work done, so I want to make sure that I thank everybody and let you guys know just how many people it takes to make all this stuff happen. Just a quick rundown. So difference in on-farm research, or what I term on-farm research versus kind of what you might think of as the small plot research. So in university trials, they're using small plots usually on the research station or out in the field. If you've hosted a trial for them, they use small research equipment to put it in. On-farm trials, we do field scale with your planters, spreaders, combines. And so we do to kind of take the small plot research and scale it up to field scale, kind of real world applications. But we don't just do split trials. So when we first started the on-farm network, we made sure that it was not going to be just splitting a field. We do replicated treatments that are kind of randomized, so that way we can run statistics on the data that we get and get better data and get a better answer if there actually are valid differences between some of the treatments that we're doing. Because when you split a field, you might see something. If you see something, you can't really know if that's a real difference or not. If it just happened to be that way, or if it just happened to yield better on one side versus the other. But split fields can be good demos to try and test things out. So I'm not saying don't ever do a split field, but don't put too much stock in a split field. Our strength for our trials comes in having multiple locations over multiple years. So when we do first year's data, often, I'll present first year's data for the projects that we do, but I don't put a lot of stock in it because it takes many locations over different growing seasons to really get a feel for how the treatments actually respond and if we're seeing differences or not. Oh no, that's unfortunate. Okay, I might have to just tell you next. Okay, so in 2023, this year, we looked at a bunch of different trials. So we continued on our foliar copper trial. So that was a foliar applied copper chelate that we put on to wheat around boot stage. And then we also continued on our second year of nitrogen rates. This is our first kind of precision ag application map, yield map, harvested trial. Oop, can you go back? Second year doing that. So looking at different nitrogen rates, and we're targeting it on high-yielding varieties with the goal of doing it over a number of years to look at high-yielding low-protein varieties and low-yielding high-protein varieties. We started looking at biological inoculants because that's becoming quite the big thing. So two things that we did this year was we looked at Pivot Bio, so Return is their wheat product, and then we also got a grant to look at Johnson seed compost extracts, if you've heard of that, and if you haven't, I will explain it later. Continued on with a rock rolling trial that came about just from some growers who had some questions about it last year, and a long-term cover cropping trial. Also worked with U of M on green seeding soybeans into a rye cover crop we've been doing for the last couple of years, and so you'll hear more about that from Angie Pelletier and Lindsay Pease later today. And then we're still continuing on with some on-combine protein mapping and looking at volunteer wheat as a cover crop, but we won't be going over those data today. Oh, and one more thing. On the back of your guys' agendas, there is questions, just a little survey. We're always looking for research needs, research questions that you guys have, but this year it's particularly important. We added some kind of announcements and a little discussion, research needs discussion at the end of the year. This year it's going to be particularly important to get your guys' ideas for projects or concerns that you have on the farm. So throughout today, if you have any ideas of things that the Wheat Council should be researching, whether it's with the on-farm network or with their university small plot lease research, please scribble those down on the back, and you can leave the agenda on the table and we'll collect those afterwards. All right, so moving on To foliar, getting into the data. So foliar copper, this will be the first section in your books if you want to open up.
It's just the data.
Of course, the clickers were working perfectly this morning. And then when it comes to showtime, they don't want to work. All right, so if you want to go to the next slide. All right, so foliar copper. So our objective, this came about because some of our crop consultants and growers in the area had been noticing copper deficiencies on mineral soils. So copper deficiency is pretty common in wheat on a peat soil. I think peat just ties up the copper, and so it's pretty normal to apply copper in a peat soil, but they'd been noticing it popping up on mineral soils, and so we wanted to do a little bit of research looking at copper applications on, regular valley soils. And so what we did, so the product we used was actually donated by Helena. We used Elemax, which is a copper chelate, and we were targeting fields that were testing below 0.4 parts per million copper, and so that's kind of the threshold of when ag advice and the literature usually suggests that you'll see a response to a copper application is when you have a soil test that's below that threshold, and it usually tends to be the sandy soils. And so basically we just did copper, we just took the sprayer out at 4 to 5 leaf stage, so right about the same time as you're doing your herbicide passes, but we separated it from the herbicide pass. So with this, we didn't tank mix, this was a separate pass. We did it tillering, half a pint of Elemax per acre, 20 gallons per acre. Just treated and untreated. All right, so next. We had 3 locations this year in 2023. We had 2 last year, one way up in Roosevelt, and then one in Beltrami last year. And so this year we had a site in Argyle, another one in Beltrami, and then one in Gently. And next. Oh, and I just wanted to point out on that last one there, so we didn't have a soil test from Argyle, but all of the sites that we were using, we were targeting tests that were, soils that were testing low or less than that 0.4 parts per million threshold on their soil test. So in 2022, just to recap the data from last year, Beltrami saw a little bit of a yield bump. Actually, it was like a 4 bushel yield bump. Roosevelt didn't see anything. Roosevelt didn't get any rain last year, so that was kind of expected. But again, just 2 sites in 1 year. So going next to this year, actually, go to the next slide too. So next to this year, we didn't see any significant differences. It was kind of, and I think in the last slide I was trying to highlight, so just a reminder for everybody when we're looking at LSDs and CVs on the bottom of all those tables, you might be familiar with those. If you're not, you're usually on the bottom of all the yield trials that you're looking at, especially the university yield trials. So LSD, least significant difference. So when that number is there, that's the minimum difference in bushels that 2 treatments need to be different. And that's just some statistics that we use to try and help us figure out what's variability in the field in the year and what's an actual true difference in a test. CV is coefficient of variation. We don't have to get into it. Joachim would probably get into it. You guys just need to know less than 10% is what you want to see. Over 10% kind of indicates that the data is kind of all over the place and the results are going to be a little less reliable. So less than 10% is what you like to see. Can you back up a little bit? So just to recap, let's see, so we looked at, so we didn't have any yield differences this year. Also, we added tissue testing this year. So this is the parts per million of copper. We did flag leaf at late boot, kind of early heading stage. We collected tissue samples, didn't see any differences there. So I don't know if it just wasn't a copper deficiency kind of year, or maybe, and if something shows up in patches, but not necessarily the whole field, that's not going to show up in the data either. So no differences in yield, no difference in tissue tests, so not much to show this year. So next slide. Just to kind of summarize, we did have one site that yielded 4 bushels more with one application, but we didn't have any response in any of the other locations. Last year was a little dry in locations. This year was dry in some locations. Maybe it's just not great testing years. Although the tissue tests were testing within the sufficiency range. So maybe, I don't know, take it for what you will, I guess. I know mineral soils, granular copper sulfate fertilizer applications tend to be more efficient when you're trying to correct a deficiency on mineral soils. And we were using the, foliar applied copper chelate. And so maybe if we were using, or if you guys wanted to try using the granular fertilizer to correct a deficiency, that might be a little bit more efficient, but that would be a different test for another year, I think. All right. So going on to nitrogen rates. So this trial has been I think garnered a little more interest in growers. What we're looking at, so I know we've done a lot of different nitrogen rate. Universities have spent a lot of time and money researching nitrogen. You guys spend a lot of money on nitrogen. We all know the nitrogen cycles can be complex and fickle at times. So what we were trying to do with this trial is compare yield, protein, and profitability in the response to the nitrogen response with kind of our current varieties, our newest varieties, with specifically trying to look at kind of the high-yielding varieties, the high-yielding, low-protein varieties, and then maybe comparing those to the profitability and nitrogen response with varieties that are lower yielding with higher protein. Which usually, I guess to kind of sum it up, would be like comparing what's the most profitable way to fertilize like a university variety that yields a little bit lower, has higher quality, versus what's the most profitable way to fertilize, like a trigger or a buster, something that's really up there but has low quality or low protein. And so for the last 2 years, we looked at rates of 0, 60, 90, 120, 150, and 180 units of nitrogen in the field. So on the next slide is kind of a picture. So this is our application map, yield map trial. Easiest one for growers to do yet, because they don't have to do anything different other than calibrate the combine at harvest. And so we wrote each of the 6 rates into 3 or 4 applications. Those blocks are about 1 acre a piece each, so that we do have to have a 0 rate in there, but in total it's only about 3 acres of the whole field that are going to have a 0 rate for this trial. And so we write that in. The spreader is able to spread it, or if you go to the next one, or we did anhydrous on one site this year. And then we get the application map just to verify that it went on right, or verify where the plots went. And then I can use that application map to trim out the yield points at harvest. And then so we can average those yield plots, the yield data points within each plot to get our data that way. But the protein is always the kicker. So why don't we do this for every trial? Because that would be really awesome and easy for everybody. Is always getting that protein sample. So what we have to do to get the protein sample is hand sample about a week or so before harvest. Once the wheat's dried down enough, but before you guys get out there with the combine. And so it takes basically 2 people, 1 person cutting, 1 person holding a grocery bag to stick the heads in. And we follow the application map and just cut heads continuously as we're walking in a line or zigzagging through each plot. Which it only takes about a couple hours per site, so it's not terrible. But could you do it on 50 sites a year? Probably not. And so last year we had 2 sites. Sorry if it's ringing. If the ringing is getting really bad, we can fix that during the break if you guys are having problems with that. Last year we had 2 sites. This year we had 5 sites, which was really great. Get lots of extra data that way. So we had Goodridge, one around Goodridge, Red Lake Falls, two kind of around St. Hilaire, and then the one in the Terrebonne, south of Red Lake Falls, Terrebonne area. And so that's, you guys have the field info. You can see planting dates, harvest dates, and everything there in front of you. Majority of the sites, everybody was pretty normal on residual nitrogen, so nothing too terribly out of the ordinary there. So next to mainly looking at the yield data. So I think I might go through everybody's fields individually. So started over at Goodridge. This year was I don't know if it was a great nitrogen response here. I think a lot of people saw that. I think kind of the story for this year was a lot of the wheat picked up that deep nitrogen, deep soil moisture to yield. For the guys that yielded really well and got it, the wheat really went deep for the nitrogen and moisture, which doesn't help when you're trying to do a nitrogen rate trial. So here at Goodridge, we did have significant differences in yield and protein. Protein tended to increase with fertility like we would expect. Yield, oops, I was looking at Red Lake Falls. And yield also tended to increase with fertility like a person would expect. But actually, when we're looking at partial profitability, that's kind of the main thing that we're going for with this trial. And so I calculated everything at 750 wheat and 550 urea, if that's kind of in the ballpark where I think at least spring prices were anyway. So you'll have to figure that out with your own prices. Let's see, but at least at Goodridge, the overall highest profitability was at about 120 pounds. And I think the story with most of these sites is that between 60 and 120 pounds was actually your most profitable nitrogen rate this year. So let's go to the next slide, looking at Red Lake Falls. This was on Rothsay, so Goodridge was on Rothsay, this was on Rothsay. Rothsay seemed to have a pretty good year. I think Goodridge had a little bit more rain. This Red Lake Falls site didn't quite catch quite as many of those rains as some other people did, more to the north. And so we had some significant yield differences, but it was really mostly just fertilized was different from zero. So there wasn't a whole lot of difference in nitrogen rates after 60 pounds. Although the nitrogen was there to pick up in the proteins, the protein increased with all of that extra nitrogen. And no differences in tissue nitrogen. Tissue nitrogen was everything was collected late boot, early heading. The ag sufficiency range is like 3.5 to 5 is your sufficiency range, and everything over 5 is high. Most of the sites, I think, tested really high for tissue nitrogen, so I think nitrogen was there. I think water was the limiting factor for the most part in some of these locations this year. So like I said again, probably most profitability-wise was about 60 to 90 pounds there. So next slide, looking at St. Hilaire, our first St. Hilaire site. This was on Murdoch, and so kind of similarly to what we just saw with Red Lake Falls, the zero was significantly different from the rest of the treatments, but then the rest is right there in the 80s. So we didn't see a whole lot of increasing in yield or protein there with increasing nitrogen rate. But we did have one, for some reason that site at St. Hilaire had super high residual nitrogen with the 180 pounds of nitrogen, but not any of the other treatments, so it wasn't really a gradient like you would expect. So that was a little odd. I don't have a good reason for that other than it was a lot of fertilizer. Okay, let's look at the next one. Second St. Hilaire site was a little sandier, I think. So this site, no significant response to yield. Everything was kind of right in the 60s, 70s right there. I know this site also had another trial right next to it. I don't know if kind of what I'm getting from this grower, I don't know if maybe the nitrogen rate was maybe just a little too high. So there wasn't, or the soil was supplying all the nitrogen that it needed for this year. So it wasn't really a much nitrogen rate response for this trial. Protein did increase a little bit with nitrogen fertility, and this is on trigger. So trigger is one of those generally really high yielding ones, but lower protein ones. So kind of what I've noticed even with the trigger candidates, some of those LCS hybrids really tend to stick around 10 to 12 protein. It's really high. Even with 180 pounds of nitrogen, you couldn't get the protein any higher, which if it didn't have the moisture to get the nitrogen into the plant either, that doesn't help either. So no differences there either, unfortunately. So I'm sorry, this isn't a little more interesting with more differences in yield. So again, looking at your most profitable nitrogen rates, there wasn't any significant differences in profitability, but what do we have? 90 pounds there is the most profitable when you're calculating your prices in. All right, so last site at Terrebonne. So this was kind of southish of Red Lake Falls towards Terrebonne. Again, we had differences in yields. So this site had about 8 bushels, was a significant difference. So pretty much everything was different from 0, but again, not a lot different after 60 pounds. And so the most profitable rate, while it wasn't significant, just looking at the numbers, your highest profitability here was at 120 pounds. So between 60 and 120 pounds of nitrogen. Okay, so just to go to the combined locations. So the nice thing about having lots of data points and lots of locations, as Joachim would always say, is that it makes it a lot easier to do statistics and get some significant differences. So when we look at everything all together, so this is 22, and 2023, so all 7 locations that we've done so far. We got significant differences across the board, but that doesn't mean that every treatment is different from each other. So when we're looking at the yield specifically, about 6 bushels difference between treatments. So that 0 rate still averaged about 60 bushels of nitrogen, which mostly comes from this year's data, which isn't bad for zero nitrogen. We just tend to increase our yield with nitrogen. Not as quite a big response as we would normally have expected, or as I would have expected to see, but the most profitable, even when we're looking at combined locations, the most profitable nitrogen rate when looking at everything together was still 60 pounds of nitrogen. Again, last year was a little dry. This year just wasn't a good nitrogen response year. And so I know if you paid a lot for nitrogen last fall, I'm sorry, this year. I don't know that it really, all the nitrogen paid off this year, but what do? You can't predict the rain. And so we'll continue this trial on. I think it'd be really, like I said, it's really good to get extra growing seasons in there. So this year ended dry. Granted, I know everybody had a lot of good yields. I shouldn't say everybody. A lot of folks had a lot of good and high yields, and I think that really was just from the wheat roots going deep and pulling a lot of that deep soil moisture, deep nitrogen from below 24 inches, which we don't really measure. And that really fed the crop in the areas that got the moisture, and some areas just didn't get the moisture. And so we'll keep continuing this on and try to focus on, also collect some other varieties in there, maybe get some lower yielding, high protein varieties there to keep, so that way we can start making some comparisons between those two. And I think I kind of covered that, so I think we can switch to the next one. Does anybody have any questions? I forgot to ask about questions for copper. Does anybody have any questions about the nitrogen rate trial or anything that we've been seeing or anything you guys saw this year? All right, well, yes.
I got one, Missy. So just to clarify, the treatment applied pounds of, that's in units of N.
Yes.
The soil test was additional to that.
For the pre-trials nitrogen?
Yep, for these trials,
That would have been all included, so I think total we were trying to shoot for
okay,
180 pounds of nitrogen.
180 total with the soil.
With that high rate, yeah.
Okay.
Oop. Could you say the question again?
So it says here treatment units. So I thought that would be applied on top, because if you have 60 pounds already in the soil, how do you reach your 60-pound nitrogen rate? That's a frequent problem for us.
Oh, yeah, no, that is, I guess, yeah, that's maybe something I didn't factor in, was like trying to hit that zero rate if you've got 60 pounds of nitrogen in.
So I'm thinking it has to be applied on top, well, I'm not sure, but I'm just wondering what's the difference between the 0 and the 60?
You know, I think you pointed out something that I missed, so that that is a very good point, yeah, to throw into there. So I think, yeah, at that site, the 0 and the 60 probably would have been the same. Okay. I'm racking my brain. Sorry, I'm trying to think on my feet here, you guys. Okay, so one of our sites mentioned that I think when we did I think I told you guys wrong. This would have been units, Tim, this would have been units applied on top of soil N. Sorry. I think my original intent was to incorporate them all together, but when it came out to getting it on, it was just additional units applied. Okay, I'm sorry. Forgive me. I told you guys wrong the first way. So total, the treatments applied would be on top of your soil, residual soil nitrogen.
Okay, yep, that makes sense.
If that makes more sense.
Yep.
I'm sorry, I mixed that up when I was talking to you guys.
You're
So it is
forgiven.
good to correct me. I am open to correction because I am definitely not a perfect person.
One more
All right,
question.
one more question.
I have another question. Did you have to do the same pattern for the flag leaf collection as the protein?
Yes, so the flag leaf tissue samples would have been collected in the same manner as the protein samples. Was it in the exact same steps in place? No, it wouldn't have been in the same step, exact same, footpath, but we would have followed a similar method of collection. It would have been the same method of collection.
Missy, one more question.
Yes.
For those that aren't as well-versed in the different varieties of wheat, what would you say about the 7 different locations and the wheat varieties that were used? You were talking about the high yielders with lower protein versus lower yield with higher protein. What would you say you had over the 2 years?
I think, for the most part, I think we got more of the racehorse varieties. Rothsey, actually, I was thinking it would be a little lower yielding, more like a Linkert, but it actually did pretty good. Actually, what I would really like to see is more, have some Linkert sites in here to compare, with like Linkert to like 9590 is pretty balanced, but like some of the LCS varieties like Trigger and Buster, those would give really good contrast. So we had, Rothsey, Murdoch, 9590 at Trigger, especially Rothsey, and I say 9590 tends to be pretty well balanced actually, but Murdoch and Trigger would be more of those. I would categorize them as the high yielding, lower protein varieties. And then, I don't know if anybody's still growing Linkert, if Linkert's finally growing by the wayside, but that would be definitely what I would, target as a lower yielding, high protein variety to compare it to the others. Linkert and similar equivalent varieties. All right. Any other questions?
Want to make one other quick comment on this one, Missy. So this trial in particular, I'm Tim Osowski. I'm on the advisory committee of the On-Farm Research Network, and I think one success of this trial shows how we can use yield mapping to speed up some of the processing of the data. And with this many treatments applied, it shows how we can do that on a larger scale in-field grid system. Of course, getting the protein sampling done still requires a lot of labor from, so Missy put a lot of time into the sampling of these, but that's still going to be a complication of any wheat-specific trials going ahead, is getting that protein sample. And the method of doing this was to hand collect in each of these grids. But I just wanted to make that comment. I think that's a success of this trial in particular, is implementing the yield mapping into it.
All right. Yes, thank you, Tim. Yeah, and before we move on, I'll just mention just for everybody to individually, think about your costs and what you're usually yielding on your farm. I know weather plays a huge impact on how your nitrogen rate's going to respond, but just look at your nitrogen rates and consider, if you're maybe if there is room for you to try pulling back on fertilizer, if you might be over fertilizing a little bit, or if you're not think about your rates, or if you're not fertilizing enough too. So just you don't have to do a full on-farm test with us. You can try putting in some strips at home too, to kind of just take a look at. It's always good to assess and look at your practices. And so even if you're, trying a few test strips, just to kind of verify and validate your practices, especially when it comes to fertilizer, always, do your own testing too on your own ground, because everybody's a little bit different. So don't be afraid to try something.
Sorry, one last question. This is excellent data, by the way. Do you know how these results compare to years past?
Years past as in?
At these locations, or what you would have expected based on historical nitrogen response data from the region?
So how do these sites compare to years past? That I wouldn't know. I think I'd have to sit down and talk with each farmer individually, just what they usually typically expect to see on these fields on their farm. I don't know that they have kind of past nitrogen rate response history. I guess it would depend on the farmer too. Is that what you were getting at?
Yeah, sorry, go ahead.
You had one last year, correct? And I remember, I think it was in East Grand Forks, but I
Okay,
feel
you just
like,
meant as compared to last year, this year compared to last year?
I think you mean overall, but last year you did have one, and that was kind of similar results. That I remember the excess nitrogen didn't pay for itself.
Yep. So one site, the nitrogen didn't pay for itself. It responded kind of similarly to this year. And one site, so I think the East Grand Forks site from last year didn't see a huge nitrogen response to increasing nitrogen, and the Red Lake Falls site yield kept climbing. That was kind of a normal nitrogen response curve that we're all taught about, where yield and protein kept going up with nitrogen. So that last year we had two contrasting sites. I think rain usually plays into that, rain and soil type. One was in the valley soil and one was in sand.
Thanks.
Alrighty. And there's always room for more questions. If you think of something, you can write it down or hit me up during the break, or right before lunch if you can sneak it in. Okay, this year was new. Do we have, I don't know if any of the Pivot Bio reps are in the audience this morning. I know Pivot Bio, they'll for sure be here tomorrow. I don't know. I think they were going to try and make it today, but I don't think they made it. So, biologicals have definitely exploded, to say the least, in the last year or two. I think we went from 4 years ago, nobody really talking about it, to now everybody and their brother has a bug in a jug you can buy. So we looked, but I think a lot of growers asked about Pivot Bio, and that one they've been doing a lot in corn. And so they were willing to donate some of their product to test on wheat, so they have their wheat seed treatment. So I'm not endorsing them by any means. We're just testing to see if it works. And caveat, I will throw out there that I did make sure that they would let us, before we even started the trial, I told them I wouldn't do it unless we could share data, even if there wasn't a response. So I'm not beholden to Pivot by any means, but also a very interesting thing to test. So Pivot Bio has, I believe it's an engineered organism, which kind of makes them a little bit different from some of the other things that you can buy from other companies. They're taking organisms that are just naturally found in the soil and sticking them into a product that you can buy, whereas I think this one they did a little bit of gene editing. You'll have to double check with them and ask them how exactly that works tomorrow at their booth. And so we just wanted to test, how much fertilizer, if you can replace any amount of fertilizer with the Pivot Bio. So Pivot Bio is a nitrogen fixing bacteria. And so our treatments with this was just doing the 100% producer's nitrogen rate and then knocking that back 25 units of nitrogen, either with and without the return applied, or the Pivot Bio. Their wheat chemical is return. And so the idea, you can go to the next slide, the idea being just seeing if we can use the pivot to replace 25 units of nitrogen. So we had 4 different locations. The Pivot Bio guys were really actually really great in helping get this trial done. They came on that first picture there that was on the screen. They came and brought their mobile seed treater out, and so I think May 11th we were able to run around to everybody's farms and treat everybody's wheat seed. They did that for us, so that was really helpful. They also took samples of everybody's wheat seed after it was treated and got it tested to see, make sure that it was to see how many, CFU is colony forming units, so see how many organisms were alive and present on the seed. And so they have a 2-week application window for their product, so they want you to plant it within 2 weeks, is the kind of the longevity of the bacteria on the seed. And so all of our sites got planted within a few days of treating, except at Mentor, I think they had some, planter issues, so they got delayed a little bit, but they were still able to get it planted about 10 days. So still within their 2-week window. So I think you can see there's a second sampling. The rep actually came out and sampled their product again, or their wheat again. And you can see the bacteria went from like 6,700 to 720. So I think just good for everybody to know that, the longer you wait to plant, that those, your bacteria counts are gonna start going down. But I did ask the rep what he thought, if that negated any response, and they think that, I guess their policy is that that's still okay, that, there's still bacteria there. Once it gets in the soil, they're gonna start replicating, they're gonna start reproducing to recolonize the wheat roots as they start sprouting. And then on the last one, unfortunately, the lab lost Terrabuns, so sorry, they lost your samples. All right, so the next one. Again, you guys have the location data there, just looking at everybody's planting dates, seed treatments, field rates. So Pivot Bio, their policy is that you can have your seed treatment, standard seed treatment, so fungicide and insecticide underneath the pivot. So they said you can put the pivot over the top of your seed treatment, but they don't want the pivot underneath your regular seed treatment. And so we had 2 sites that had seed treatment underneath the pivot, and then 2 sites that were just bare seed. All right, so next, what we saw, so looking at Mentor first, we didn't see no significant yield response in Mentor. It was a little less rain, I think there. Unfortunately, I usually get all my rain data from Climate Field View, and Climate Field View kept telling me that all of our sites got 30 inches of rain this year. So I lost my easy source of good rain data, or rain estimates anyway. So hopefully when Daryl talks about his end-on stations tomorrow, he'll have some good news about them expanding the end-on network into Minnesota, so we can have some better rain sources for you. So, but back to Mentor. So yeah, no real response here. Next site, in Stephen, I think it was actually our best field. No significant difference in yield, which can be a good thing. Slight difference in residual soil nitrogen, but go to the next one. I think the yield was the one I was focusing on. It wasn't a significant difference, so you got to take it with a grain of salt. But I will say that just when we were harvesting it, the reps were very consistent in the minus 25 pounds N treatment yielding like 2 bushels less. So it's very hard to get significant differences with just a single on-farm location. There's just not enough data points for the stat. But I think there was a trend to be considered here at this site. So it looked like something was going on. I just can't verify that numerically. At St. Hilaire, again, no differences in treatments. I think if you scoot ahead just to focusing on the yield, we had 80, 82, 80 and 82. There wasn't a nitrogen response there, even with the minus 25 units. So I think, I don't think we just didn't have a nitrogen response there, regardless. So if the minus 25 units of nitrogen was enough to maximize yield, the pivot wasn't going to add anything on top of that. So I think that might have been just soil and fertilizer supplied more than enough nitrogen there. All right, and then at Terrebonne, we did have, it wasn't a significant difference, but there was a bit of a yield difference there with minus 25 units. So I think we did have, definitely did lose a little bit of yield. It tended to lose a little bit of yield with our minus 25 pounds of nitrogen. They were pretty dry there. Still got 70 bushels, but not a lot of rain there. And so rain plays into nitrogen cycling, and then even more so when we're looking at biologicals, because we've got living organisms that we need to keep alive and living in the soil and keep them from going dormant if they're going to work. So I know that site was drier this year, and so I think kind of same as with nitrogen, when you have dryness, you're probably not going to expect as quite a much of a response. So there, I think we can move to the next, combined. Looking at combined locations, again, no huge differences in yield or big responses. I'm not 100% sure of the cost. I don't know I'm not sure if when we started it this spring, if they had a cost for their wheat treatment yet. So I can't tell you if it's going to be, cheaper or more expensive than 25 pounds of nitrogen. But I think we're looking at 1 year of data. It wasn't a great nitrogen response here. I think you can skip to the next slide. Yep, the summary. I think more years of testing would be good to include, especially if we can get some environments where we do have more of a response to nitrogen, a little bit better rainfall year, to start picking out when or where or how much you might expect a response with biological testing, not just pivot, but biologicals in general. Yes, go ahead.
If you had 30-pound ranges on that last study that weren't showing any response, it seems like with the Pivot Bio, you should have been comparing it to a 0 application versus just a treatment with Pivot and see if there was any response there, because, we already looked at that last study that showed there really wasn't any differences in even 30-pound rate differences.
Mm-hmm. Okay, yeah, so the main question is, why not have a 0 nitrogen plus pivot, And I think the tricky part there is, well, for one, I They wouldn't let us test zero nitrogen because they knew that the biology isn't going to make up for you just having, zero nitrogen out there. So I think at that point it would just devolve in, if you had a zero nitrogen with and without pivot, it would devolve into a nitrogen rate trial of comparing with and without nitrogen, because I don't think that biology it's never going to be able to supply all of your nitrogen needs. And without doing a huge small plot trial, it would be really hard to pick out where exactly it's going to fall, how much exactly how much the biology would be able to supply for nitrogen. So I think we picked the 25 units because that was a conservative estimate, to estimate, okay, can we at least get 25 pounds out of it or not? I think you'll see with the next trial that we did do the 0% and 50% and 100% nitrogen, and that wasn't the greatest treatment option. So I think you'll see why we probably shouldn't do that in the next trial that we talk about. Go ahead.
Was the 100% nitrogen rate based on state recommendation?
That would be the producer's it would just be the producer's nitrogen rate. And I'd say most of them are probably just running off their AgVise, their soil test recommendations.
Okay, and what's the lowest rate of nitrogen that you are allowed to use in this trial? That people buy
The lowest rate? I guess we didn't put a cap on, what's the minimum nitrogen response or nitrogen rate that a grower would need to use. Are you saying like everybody would have to apply X amount or higher?
Like for instance, at our location, the recommended nitrogen rate for our state always overshoots. And so it does come with a plus minus 30 pound range. So it's definitely minus 30 pounds where we're at just because of the limitations on moisture. So if we went with the 100% recommendation, we wouldn't see a difference going down 25 pounds. But if we chose a lower rate, we might.
Gotcha. Okay. So you're mainly wondering if people are over-fertilizing in general. And I think that's where having the 25 unit, it's going to be hard. It's going to be hard for us to know, especially on a year-by-year basis. I think that's where having the minus 25 control without the pivot comes into play, because we expect to see a yield drop there. I guess ideally when we see these sites respond, the minus 25 would have a lower yield than the 100% to kind of confirm that we're about in the right range for fertilizer rate. And then if the pivot was working, as everything should, then that minus 25 plus pivot would be the same as the 100% rate. So that would kind of be that's kind of our internal check for trying to verify that we weren't fertilizing too much, if that makes sense.
Yeah, so I would say the two questions alluded to it, that an application rate of nitrogen of 122 to even up to 185 pounds of nitrogen are the field nitrogen rates. It would be interesting to see a much lower rate to see if there's a response, because like you said, there's not any difference between the 100% and the 25%, and the minus 25 pounds of nitrogen.
Yeah.
So yeah, and it would be interesting to see what a lower nitrogen rate would do.
Are you guys volunteering for next year?
Potentially.
Yeah, and no, that's some good comments, and that's the good feedback to hear, to maybe target growers who are tend to be more conservative on nitrogen, on their nitrogen application rates, to I guess make sure we see kind of a nitrogen response with that 25 unit control. So yes, all good feedback and points, and of course the weather plays into it too. Sometimes you never know when 60 pounds of nitrogen is going to be plenty for 80 bushels of wheat, or if all of a sudden you needed 150 pounds of nitrogen for 100 bushel wheat.
That's a great comment, thank you. And something to keep in mind as we're going through this, everybody too, if you have an interest in one trial that was just spoken about like that, we're also looking for feedback of how to tweak these trials that are potentially done in the future. So That's a great comment. There's different ways that each of these trials could be done. So that's something to keep in mind as we're going through this.
Yes, that is one thing about research is there's 100 different ways to do everything, but unfortunately you can only fit 2 treatments into a field, or at least a field scale field. So we got to start somewhere. So yeah, if you guys have any suggestions or things you'd like to see, definitely write it on the back
Yeah.
of your agenda.
One question here.
Yes.
You know, these areas are very different soil types, so it'd be interesting to see what the organic matter was on some of these going forward, you know, just the data, because.
Did I not include organic matter in this?
No, because they're very, very different soil types, so I was just, that'd be interesting to see this too.
Okay, yes, yep, no, that's my bad for not putting the organic matter on the soil data. Very good point. Oh, a question was just wondering what the range was on organic matter. Off the top of my head, I couldn't tell you.
One other thing that's different this year with Pivot Bio is that it's on the seed versus what they'd done in the past, because we'd worked with them for about 5 years, and that was putting down the furrow. So is it as effective on the seed as it was on the furrow? The furrow application would be a pain for most farmers and expensive to put on your planter. So it doesn't bode well as a additive, in my opinion, but it works much better on the seed from a practical point of view. But is it the same? Don't know.
Lost my mic. Oh, there it went. Yeah, I think that, and there's at least anecdotal, if not published research. I don't know about published at least anecdotal evidence that, yeah, the in-furrow applications tend to work better than the seed treatments. But yeah, like you said, very few people are set up for liquid on their wheat drills, except for you guys, if you want to volunteer for next time. Yeah, so that's why I think we definitely had to go with the wheat seed treatment versus like doing an in-furrow with your soybeans or your corn. So yeah, application method definitely also going to play into efficacy with these kinds of trials also. Which in the next trial, I think we also will see a little bit suggesting that also. Any other questions about pivot before we move on?
They looked at doing it fertilizer. Pregnant pivot.
Say that again.
Has anybody heard from Pivot if they're working on an impregnation for starter fertilizer on grain drills? To see if there's any benefit of doing that to your P and K as you're using it as a starter for a grain application?
To be quite honest, I don't even know that was a thing, so I guess I couldn't tell you. Craig's always full of ideas. I can always get some ideas out of Craig. All right. Okay. With that, I think Pivot will be, and some of the other biologicals. I mean, they're good to test too. I think, like I mentioned before, having the edited, microorganism maybe sets Pivot a little bit apart from some of the other products. But yeah, I think it's definitely something worth continuing to test also. And maybe if you guys would like to see it in other crops, let us know, write that down, and we can maybe see if we can work with soybeans or corn. I guess, I don't know if soybeans would need it, but see if we can work with corn or some of the other crops. We'd have to collaborate with them on kind of funding for that. But if that's something you guys want to see, let us know and we can pursue that if that's of interest. All right, so kind of the flip side. So Pivot's something you can buy. A Johnson Sioux bioreactor and looking at compost extract, this is a biological that you can make. So I imagine probably 95% of the people in here probably haven't heard of a Johnson Sioux bioreactor. So why don't you flip to the next slide. So what is it? Basically, It is a method of aerated composting that doesn't require turning. So this is getting into growing your own biologicals. So the biological companies, basically they all got all of their bacteria and all their microorganisms from the soil. So those are, depending on different companies, they're gonna be different, sourcing them from different places across the nation. They're not necessarily gonna come from right here in Minnesota. So composting is a way to kind of grow the whole biological community. But it's going to be a local biological community because it would be made from the materials that you have here or that we have locally. So these would be native biology that you would be applying to your crops. So composting, I know we're just referring to composting when we're talking about gardening, but this is a way that David Johnson and his wife, that's why it's named Johnson Sue, they came up with to scale it up to field scale production. And so there are actually farmers kind of sparse throughout the country who are, and also there's some commercial high value crops that are using this method as a source of their fertility and biological inoculation on a commercial scale. And so, kind of like it says up there, the idea is that we're going to treat the seed with the whole community of microorganisms, hundreds and thousands of microorganisms, and not just, the handful of strains that a company puts in a jug. And so the analogy that David Johnson usually uses when he's telling people about this, he alludes it to a baseball team. And he says, when you put the whole biological community out there, you're playing with all the members on your baseball team, and you don't just have a bunch of batters all lined up trying to play baseball, or a bunch of, shortstops all trying to play, baseball. You've got the full team, your whole team members, all the different players are all working together to play the game. All right, so, and that is a picture. So up here we got pictures of the bioreactor. Go ahead. Next slide. And so this grant we funded with SARE, so the Sustainable Agriculture and Research Education Council. So this was funded through kind of a farmer partnership grant with them. And so our idea with this was, just demonstrate how to make a bioreactor. And then also we worked with UMC, the rock over in Crookston, to do some small plots. So working with Lindsay Pease was also to do small plot research to look at hopefully maybe look at a fertility aspect of this. Is any amount of nitrogen and phosphorus fertilizer, can we reduce that, or can it be supplied by using these microorganisms in the soil to help unlock some of what's there and unavailable to the plant? Can we use those biology to unlock more nutrients and therefore maybe reduce our fertilizer a little bit, or be able to reduce the fertilizer? So similar concept to what Pivot was trying to do with the nitrogen. And then we're also just, when we do this, what kind of fungal and bacterial species do we get? What's the diversity that we get when we make this compost? And then just trying to, show farmers how we did it and how it can be done and what happens when we do it. So it's kind of an out there project that we pursued. All right, so next slide. So this year, so like I said, it's kind of a large plot, small plot collaboration. So we were originally intending to have 3 different producers we were working with, but one ran into had to pull out this spring. So we ended up with 2 growers that we were working with and 2 sites. And treatments didn't go quite as planned, but we have what we have. So at Crookston, we had, and this is where, the other gentleman had mentioned just looking at the 0 nitrogen, half rate nitrogen. So that's what we did in this trial. We had a 0 nitrogen treatment, 50% N and 100% N. At Crookston, those were all treated with compost extract. There was no or no control without extract. At Red Lake Falls, and I was in wheat, in Red Lake Falls, they had two trials. We did one in wheat, one in sunflowers. For the control, we had the producer fertilizer rate with no extract, and then we had 50% nitrogen, no P, and then with the extract, and 0 N, 0 P with the extract. With the idea being that we were hopefully going to try and see how much, I think we might have been a little zealous in picking those, or overly excited in picking those rates, but we wanted to see how much, if any, the biology would be able to supply for fertilizer. And then in sunflowers, it was in-furrow extract, so it was in-furrow applied just with and without. So that was, just flat rate fertility with and without extract, with the liquid in-furrow application in the sunflowers. And so we did there's a company called Biomakers. We did a microbiome analysis of the, basically vermicast, the product, what we used as the compost extract this season, and it had over, I couldn't really put it in a table, it was a lot. It had over almost 1,000 species of microorganisms, different microorganisms in the product that we applied. So 137 different species of different fungi and 850 species of bacteria.
You should clear, so the bioreactor, we made that, but the idea of the vermicomposting, which is the worm stuff, is that it sits for basically a year before you can use it. So that's why we had to get a different compost for that year.
Yes, and I will get to that in the next slide. I'll get that and explain that in 2 slides. So then, so those were our large plot treatments that we had this year. Small plot treatments is where we were looking at the breakdown of fertilizer treatments in wheat and soybeans. So we had, 4 different rates of nitrogen and 3 different rates of P in the wheat, and then we were looking at 4 different rates, or 4 different rates of P in soybeans also. So we had a wheat plot and a soybean plot, and all the different combinations with and without extract in those small plots. And so that was where we were hoping we might be able to, pick out, find where exactly along that curve the biology might kick in. Okay, so next slide. All right, so this is, I think how this bioreactor works is a story better told with pictures. And as I was, in typical researcher fashion, putting this together last night, my iPhone put my pictures into iPhone jail. So I have some pictures, but not everything that I would like to show you, unfortunately. So this is kind of how we made these. So we made these bioreactors. We made 5 of them, but in March. Because the idea is that the composting process, to grow all these biology, we add worms to it. The worms actually add a lot of the beneficial biology. Worm castings or worm poop actually has a lot of plant growth regulators compounds in it, and a lot of beneficial microorganisms. So actually worms do a lot for the biology and the fertility of your soil. And so the idea is with this is that we're composting with the worms added in this bioreactor for a whole year to give it time to mature fully before we use it. And so we built these in March. Basically, we took a tote and used a seed bag and filled a seed bag with compost materials. We used wood chips. These were already wood chips that have been composted for a year already. Leaves, horse manure, hay, and what was straw? Cereal rice straw. Yep. And so that was our kind of composting mix to mix our greens and our browns to get our nitrogen ratio and our nitrogen carbon ratio where we wanted it. And so we shredded those. And so very much big thanks to Trinity Creek Ranch for doing this and supplying the materials and doing this in our shop in March. And So we mixed all those, got them wet. Everything needs to be wet. There needs to be moisture for the the biology to live, for the composting process to work. So we got them wet, and then we filled bioreactors kind of around these chimneys. So we use the PVC pipes as chimneys, so that way the air can flow down through the whole pile constantly. So they say that, I guess they recommend, or current guidelines for building these is that each part, all of the compost should be no more than 12 inches from air. And so the chimneys are spaced so that there's always access, the air can infiltrate 12 inches into the compost set. It's always constantly aerated. Because aerated compost makes good biology, anaerobic compost makes bad biology. All right, so you can go to the next one. And then, so as Michaela had kind of mentioned, so this year, so that we built in March, that's going to take a year, and we'll use that compost. We'll test the biology on it before we plant, obviously, to see what we have there. But for this year, in order to get started, we had to source some compost product from elsewhere, and so we used Fenid Happy Vermicompost as our substitute in for this year. I do have a little jar sample actually with me if you want to see what it looks like over the break. It's very fine, looks like fine soil. And so this is how we made it with the small plots. Of course, my pictures of how we made it with the large plots are stuck on my phone at the moment. But I can show them to you at the break. So what we did in both, for both settings, is we bubbled or used air, so a high pressure air. So I used an air compressor I used a sprinkler, basically to make a big agitation bubbler to knock the biology off of the soil. So you have to knock the biology off of the soil. You can't just mix it and stir it. It's not agitated enough. It has to be vigorously bubbling. And then we strained it first through a paint strainer, and then so Craig, our other cooperator, he was kind enough to actually build and buy all the materials for the field extraction process and filtering. And so for the field applications, we did it in a mixing cone, bubbled it in a mixing cone, and then he had a setup, a setup from Spray Advantage to filter first to 30 mesh, then to 50 mesh. And I don't think we needed to go up to 80 mesh. 50 mesh was fine, and that worked fine for the filters that were on the planters too. And so we filtered the extract several times so that way we could get it through the planter, and that seemed to work out pretty well. So that's just kind of pictures of what it looks like, at least in the bucket there. Again, my pictures of it on the planter are in my phone. And so that's how we got that's how we ended up with our slurry. So for the wheat, we did a seed treatment, similar to the pivot. We didn't have any, in-furrow setups to do a liquid application on the wheat. So the wheat we treated, Greg had also bought a mixing cone that we made into kind of just a at-home seed treater. And so we, went through, calibrated, as we were dumping from the truck. Basically, we were loading from one truck into another truck and spraying and treating as we went. Just kind of calibrated our flow rate coming out of the truck, and our flow rate coming out of the spray nozzle to make sure that we were targeting about 9 ounces of fluid applied per 100 weight as our seed treatment. And so we did that as we were mixing from one truck to the other, and that way the auger could really mix everything up. And then by the time it got out, by the time the wheat got from, one truck to the other truck, it was already dry. So we didn't have any super wetness problems there. So that was good. And then for the sunflowers, when we did the in-furrow extract, we just had it filtered out and then pumped into the planter or into the drill. So, all right, so you guys have the field info table there. So that's just looking at yeah, we can go to the next slide, or background info for our 3 sites that we had with the different treatments. At Crookston, I guess there was a significant yield response or protein response, but as you'll notice, we had 30 bushels and completely 0 response to nitrogen. So this site, I know a lot of guys got rain and did really well, and this just was one site that got no rain and did not do so well this year. So we had complete lack of response to fertility, and so therefore lack of response to any biology also, to be expected. So unfortunately, that is one where the weather kind of just screwed everything up, which is the name of the game. You guys have that every year, and so does research. Go to the next slide. All right, so at Red Lake Falls, at their wheat site, so this kind of gets back to our previous question that we had about, doing the biology and looking at the 0 and 50% N. We had major differences in yield, but really what that amounted to was, without a 0 nitrogen check with and without extract, basically I think we ended up with a nitrogen rate trial of what happens when you don't, when you cut your nitrogen rate in half or don't apply nitrogen. So I think, because we went from 50 to 60 to 70 bushels, which each of the different treatments. And I think a better treatment choice here would have been to do what Pivot had, and had to have just done the 25 units, minus 25 units with and without the extract. So I think that was a lesson learned for this year. I think we might have just gotten overly excited about how much the biology was how much fertilizer it was going to give us. We had extra products. So at Red Lake Falls, they also put down in-furrow extract with sunflowers. And this site was a little more interesting. We did have a significant difference in yield. Sunflowers should be pounds per acre. My bad. Not bushels per acre. But so it was a small bump, a small difference. But we did see 65 pounds per acre bump with the extract, and that was pretty consistent across the trial. I think we had 5 reps, and 4 out of the 5 reps showed like the bump in yield with the compost extract. So I think it looked like something was going on there, and I think it was. You can go to the next slide too. So, yeah, I was thinking about the other slide, but I think it was also kind of pointed to some of the anecdotal evidence that, order of efficacy for these biological treatments, foliar application is probably your lowest, seed treatment is next best, and then the in-furrow is going to be your best shot for trying to see any response. So with this grant, we also did tissue testing, and we did soil nutrients and PLFA testing. So PLFA is a soil health test. It's a phospholipid fatty acid assay to try and estimate the number of fungi or the amount of fungi and bacterial biomass that's in your soil. And so we worked with Lindsay Pease's lab on this one. And so for the on-farm plots, we did tissue testing and PLFAs. These are just the raw averages. These haven't been run through stats yet, but everything's it's pretty even. So it wasn't, which is also part of the reason why I didn't have to run through the stats on it yet. So we didn't see a whole much of any difference really in the tissue test or the soil biology, soil health test. And this was just done on the wheat. Unfortunately, we didn't have the grant money to do it on the sunflowers, which would have been the more interesting soil to do it on. And that's always the hindsight 2020, I guess. So this is just for the 2 wheat sites. I'm working with Lindsay on the small plot data. So we still do have the small plot. We did the small plots, soybeans and wheat. Still working with that, working on that data. I think Lindsay's crew is still trying to crunch through all the soil samples, so the regular nutrient analysis, and in soil N and P tests she's running in her lab, and so they're still trying to crunch through that, so I don't have that data for you guys yet either, for the large plots or the small plots. In the small plots, I think they kind of suffered a similar fate as the Crookston wheat site this year. They were like 30, 40 bushels. Not a lot of, wasn't much difference between treatments. So we'll have that a little bit more to show you guys for next time. But just to summarize, so this was our first year go at this. This is all kind of very brand new stuff. There's no real, there's no university guidelines or recommendations for this stuff. So it's trying to figure it out and learn as we go, and learn with the people who are already working on this also. There's a lot of farmers and smaller operations trying this stuff, but there's no real replicated research out there. And so this was our effort to try and get some replicated research going in large plots and small plots looking at this stuff. So like what we talked about, I think the nitrogen rates, I think we're gonna have to change that up for next year, or change our treatments up a little bit for next year, or at least make sure we get some extra controls in there, or go with what we did with the Pivot Bio and just have, the incremental 25 units. Smaller nitrogen or fertilizer differences to maybe get a better idea of what the biology could potentially do. In-furrow, John had mentioned earlier, in-furrow applications seem to work much better than the seed treatment. So if we have more opportunities to do and look at in-furrow versus seed treating, I think that would be interesting. And so we'll be using the compost, provided everything checks out and the biology is good. We'll be using the compost in the bioreactors that we created this season to see how that goes. All right. Questions? Any questions? I wish I had more. I've got tons of pictures for you guys. I wish I had more pictures to actually get a better, show the story a little bit better. But you'll just have to wait till next time. Yes.
So for next year, it would be nice to see a comparison. There's a 50% nitrogen rate plus extract comparison with just the 50% nitrogen rate without the extract, and also for the 0 where there's not a check without the extract, because then maybe we would be able to see whether it worked at that rate. And it sounds like a lot of work though.
Yes, no, you were exactly on. I think lack of extra controls was kind of our downfall this year. We picked these we had a whole slew of treatments that we wanted, and we picked these out because you really can only fit 2 to 3, 4 treatments in a field scale trial max. And so we were trying to pick our treatments of most interest, but I think we should have swapped in an extra control in there for sure.
And my other question, what was the nitrogen rate on the sunflowers?
I think it's in the field info table. Was it 12 gallons per acre of side dress UAN?
A little bit on that question you asked there. This field received under 3 inches of rain for the entire season from when it was planted till it was combined. And the field started out with about 18 pounds of total nitrogen. So your 0 rate is 18 pounds. The whole rest of the field, the 120 acres of that field, still averaged at 30.1 bushels, same protein, same test weight. So the whole rest of the 120 acres was your control. And in there was also a source biological product that was used with 0 difference at 3 different rates on flag leaf applications as well. So it goes to point that no rain, no results.
I have a question for you also. Could it is a liquid you end up with, correct?
Yes.
Can it be impregnated on fertilizer, or would it be too much liquid to put on fertilizer for the starter?
I didn't even know. I feel like I'm out of times I didn't even know impregnating fertilizer was a thing, although having living biology in that close contact with that, concentrated fertilizer, I feel, my gut feeling is that probably, I don't know if that fertilizer would, be toxic to the biology in that close, when it's, trying to live on the fertilizer versus being separated, when you're planting in the soil.
Yeah, so if you have access to to manure, what's the, why would you go through what's the difference of going through this process versus just spreading it with a manure spreader?
If you have manure to spread, you are very lucky.
Yeah, but I'm just trying to, because this seems like a lot of work and.
So I think, so where, can I ask where your farm is?
Oh, Langdon, Walhalla area.
Langdon, Walhalla, okay, gotcha. So at least you guys have access to manure. I'd say the vast majority of the farmers, at least up in this northern area, don't have access to manure. Just by this, we don't have a whole lot of. So the composting, I think the difference would be in the composting process of, you're growing in different kinds of biology than what might just be in your manure. So it would be taking, manure is going to be part of, your feedstocks for this. So it would be taking that and scaling it up and adding more to it. But if you're even just having manure to, just spread as a base fertilizer, that's going to be extremely helpful for your organic matter and your soil biology just by itself. And this would just be, if you wanted to go through the extra work of it, would be a way of maybe also scaling it up too. So, we use 2 pounds dry product per acre equivalent, essentially, is what we applied. And so one of those bioreactors is going to create enough compost to be able to cover, 1,000 acres, if in furrow, or seed treatment for sure, maybe in furrow. So I don't know how limited you might be on space-wise, how many acres you can cover with your manure. So this would be a way to also, get it across your whole operation also.
I would also add your manure system is usually anaerobic. I mean, unless you're out there turning your piles. I know we're not. And so the idea is that in an anaerobic system, you're actually growing more pathogens or not usually beneficial organisms. The vermicomposting has aeration in it that it's supposed to be aerobic, that you're building your beneficial biology because they are the ones that thrive in aerobic conditions. That's the idea, anyways. When it first, so we mixed it all up and it probably heated to 120, 130 Fahrenheit. 150 would be the dream to, kill your weed seeds.
160 is your target for,
or maybe, yeah.
For killing weed seeds. I thought we got close. I know I have it somewhere.
I don't know. But it was maybe 2 weeks max, and then it came back down to ambient. It's in the shop right now in the university store and the other ones. What? In a heated shop, yeah, frozen probably wouldn't work. We did also add worms after the temperature came back down, which was kind of a cool experience to buy a bag of worms and see them all. So, and the worms are still in the one that we have on the shop.
So let's say you put a ton of this compost in that bucket, how much liquid do you get out of it?
So I didn't put our rate in the book. So how much liquid, it depends on how you want to mix it. So we took the kind of what, so for the sunflowers, for instance, kind of standard, what you were applying was 6 gallons per acre of, what you're usually applying with your liquid. And our target was to hit 2 pounds of the dry compost equivalent spread per acre. So what we did was basically mix what the 2 pounds per 6 gallons per acre, 2 pounds compost to what the equivalent water would be for 6 gallons per acre. And that's kind of so far it's the recommended rate, but it's the untested recommended rate for some of this compost extract stuff. So as far as your what rates you're going to mix it at, that would be some very interesting university research that hasn't been funded yet. That would be a next step for somebody somewhere. So I guess the way I would think of it is looking at, if you're using like your dry flowables for an herbicide, like mixing that up, can I compare it to that? Like if you have, I know nobody uses that, but like just for taking your dry and then mixing it up to your equivalent of what you want to apply per acre. Is that a fair comparison?
So this year you used a commercial product, correct?
Yes, it was vermicompost or vermicast, because essentially what you end up with more or less in a bioreactor using those worms to do the composting is vermicomposting and a bunch of worm poop mostly. So that's what we bought from a commercial worm operation to use as a substitute.
So why the poor ratio? Is that as advertised, having many more bacteria than fungi?
I guess I'm not sure. I'm not sure what the vermicast or vermicompost ratio is supposed to be.
Okay. I just always thought to have a good composting system, you want to really have a lot more fungi than bacteria.
Yes, no, and that is a good point. And I think part of the reason for doing the static composting with the bioreactor is so that you can allow more fungi to flourish, since you're not mixing it and breaking up all those fungi to get more fungal mass in the compost. And so that's a good point. So maybe that'll be, I think, interesting to see how, when we do our microbiome test on the stuff that we made, how that compares to the stuff that we bought this year. That is also a very good point. Any other quick questions? Or we got one more trial to go through before the break. It's definitely something new and out there, so that's why we're trying it, so you don't have to. Or experimenting with it and make all the mistakes first. All right, so our next, we have 2 trials. Okay, this one's short. I'm gonna speed through it. Rock rolling wheat. So this came up just because a consultant and some of his farmers, mostly in the Red Lake Falls area, had asked about rock rolling wheat. They were rock rolling their soybeans and they started rock rolling their wheat. And so, does that have any effect on wheat yield? We don't know. So we did a couple sites of it. We did one site last year in Red Lake Falls. The other sites kind of fell through last minute. And so this year we had 2 more sites go in. So we've got 3 sites total from this year. And this is pretty simple, rolled versus unrolled. So planted, roll, and then rolled before the wheat emerges. You can go to the next slide. And this, for those of you who are in the valley, this probably is of no interest to you, but for those who live in rocky areas, it's a little more pertinent, especially for those folks who are also doing grass seed and interseeding their grass seed in with the wheat, and who want to who have to maybe also, pound the rocks in for, trying to protect the combine from, when you're doing wheat and grass seed at the same time too. So we had 3 locations this year. You got that info. We can go to the next slide. So we had Crookston and Dorothy this year. Crookston actually saw a small yield increase this year, or 3 bushel yield increase this year. Dorothy didn't have any differences in yield. We can go to the next slide. And then Red Lake Falls, the site from last year, also didn't have any differences in yield, just rolled and unrolled. And so I was able to combine all those and look at that, no real differences in yield. Honestly, we didn't expect any, but I think the point of this, doing the rock rolling trial, was just to, I think, verify that, make sure we weren't injuring the wheat in any way, affecting stand. So kind of summarizing, one thing the consultant noticed, and we did notice last year, there was more soil crusting in the rolled treatments, which, you guys see that in the soybeans also. Crusting happens a little more, and I think the consultant who did this trial, his theory was that the crusting had actually helped keep some of the soil moisture in, and he was thinking that might have been why we actually got a little bit of a yield boost. just kind of made an extra little barrier layer from the soil evaporate, or the water evaporating out of the soil. So that, and I think we did notice the crusting last year also. Didn't seem to have an effect on yield. But, so, very simple, straightforward trial. Only 3 locations. I don't know, whenever you've got soil crusting, it didn't seem to have an effect on yield in these sites, but I guess take that with a grain of salt, because generally we don't want crusting. It might have helped this season, but that can impede your emergence, affect your stand, affect your water infiltration in other seasons also. So, take that as you will, if you happen to roll rocks. All right, so that was just a quick, simple trial. So our last trial that we want to cover today is one with the Soil Health Partnership. And so
I'll give a little backstory for her.
Yeah, so
Since it kind of started before Missy. I'm Michaela Tabbert. I'm on the Minnesota Wheat Council as well. Probably 6 years ago or so, my family was approached by Dave Torgerson and kind of they were searching for sites for the Soil Health Partnership, which was a partnership formed a lot by national corn growers, but the National Wheat Foundation, and they had a lot of nonprofits, and it was a kind of a collaboration to test the soil health practices on on-farm scale across the nation. Probably a year or 2 into us starting the trial, they kind of dissolved the Soil Health Partnership and dedicated funds elsewhere. The trial that we had was a 5-year cover crop versus no cover crop field trial. And the setup was, randomized complete block. And the idea was that we would be able to farm the treatment separate. That was kind of how they set it up. So we put it in 100-foot wide so we could possibly spray them separately. I mean, it's hard to plan for 5 years. But then Soil Health Partnership was dissolved, but Minnesota Corn was willing to pick it up, and so through their innovation grant program, they've continued to fund it. Missy has been contracted to kind of help run the stats and just management and data collection. So that's kind of history of that experiment. This was new ground to us. We're no-till for the most part. We used to do some strip-till. So it was maybe in our rotation 1 year before the trial, so it hadn't really gotten much for cover crops prior to that. And it was very simple, like cover crops every year on the cover crop treatment, whatever that looks like in the crop rotation versus no cover crop. So you can go to the next slide. So yeah, that objective, they were trying to assemble a full database across the nation of comparing a lot of soil characteristics. And yield and profitability was the goal of that. It would have been really neat if they would have filled out that data across the nation. I think it would have had a lot of value. So we usually do diverse cover crop mixes. Our row crops, the corn and the sunflower, and actually the year we did soybeans, we interseeded a cover crop into that. And the wheat years, it was seeded after harvest. You can go to the next slide. I won't take a lot of time with how the cover crops were seeded, but we are set up to drill 2 rows in between the 30-inch rows of cover crop, or in season of the corn or the sunflower, usually at that V4 to V5 on the corn, V2 to V4 on the sunflowers. To drill that cover crop in, we've got a twin row with a single disc opener that we do side dress nitrogen with as well. So 2 rows in between each 30-inch row. And then there was a lot of soil sampling at this. When they set it up, it was on 1-acre grids, and it was, 0 to 2 and 2 to 6-inch samples. And then they also did the Haney's at Ward, and then Cornell, which we haven't redone yet. So the idea was that to see what the difference was in soil after the 5 years. So this was the 5th cropping year, but we haven't done the final soil sampling. That will be done in the spring. So we haven't done the Cornell soil health ones again. So Missy did the cover crop strand and weed counts and biomass. Next slide. This is kind of the cover crop mixes that we used and the planting dates of them. Sorry, this isn't the cover crop yet, but this is the crops, the rotation. So corn, soybean, wheat to sunflower to corn. You can go to the next slide. Those are what the actual cover crop mix was. As you can see, we like to try a lot of species at low rates. Some of that is experimental, trying to figure out what will work well in that interseeding mix, because shading and crop canopy really affects stand. 4 out of the 5 years, the cover crop did pretty well. Even that interseeded soybean, we don't do that normally. It was just a year that those beans didn't get very big. It was 30-inch rows, so we actually got some cover crop to grow and establish in that. But that's a pretty rare year that you get that. 2022 last year was probably the hardest cover crop year. So after wheat, we had done the cereal rye and kind of had a planter calibration as far as that and put down a lot of rye. As you know, 2022 was super wet in the spring. We didn't get it seeded until the 8th of June or something like that, and we left that rye and planted it green. I believe we would have terminated like the year before, or the day before we planted the sunflower. And so bottom line, there was a lot of rye biomass, which we thought would be a great thing. But the sunflower didn't do well with it. It didn't handle it. It was solid seeded rye. And as we've progressed, we've switched to paired row rye to kind of leave a space for the sunflower. It was a lot of biomass. It really held back the sunflower. It was probably a good growth stage behind the whole time. There was some stand loss. There was also some cricket and grasshopper pressure that did very well in that biomass. And so the cover crop didn't do very well. It was pretty much eaten by the grasshoppers and crickets. So that was, it's still a system that we're trying to figure out. And I feel like we've been doing it for a few years. There's just not a lot of information on cover crops this far north, obviously. So, I'll hand it over to Missy to fill in the research part.
Okay.
Oh, yeah, you have Mike.
Yes, no, thank you, Michaela, for giving that good background on kind of what this project is about. So you can switch to the next slide. So just looking at the data from the last 5 years, we didn't see a whole lot of differences. Corn in 2019, no differences in yield, same with the soybeans. Next slide. Same in wheat, no real differences in wheat. And then, as Michaela was just describing, the sunflowers, the rye competition in the spring stunted the sunflowers, and the sunflowers were super behind all year. And you can see that definitely reflected. It was reflected. The stand counts were down, yield was down. You can see that in the moisture also at harvest. So it was a learning year for the sunflowers. And then next slide. Yep, next. And you can see, and then that carried over into, assuming that carried over into the corn this year. So we had significant difference in corn. The cover crop plots did, 20 bushels better than the non-cover crop plots, but we're assuming that was from last year. Then the cover crop plots didn't have had far less sunflower biomass going down. So those control plots had a lot more sunflower residue, which we're assuming impacted the corn and probably tied up some nutrients too. So we're guessing that's not a cover crop effect, that's a residue effect there. Just preliminarily looking at some of the cover crop data and the soil data. So last year, and unfortunately, we only have, at least for biomass and all this cover crop metrics, we only have the last 2 years because we really didn't get anything from the previous 3 years when it was actually the actual soil health partnership people managing the plots. So for at least the last 2 years, last year we did measure, differences in soil temperature at planting because of all that rye residue. Huge major difference in sunflower stand because of that rye residue. Next slide, moving to this year. We did cover crop counts. And so you can kind of see a picture. There's the quadrat that we took in corn. So it's a 2 foot by 2 foot quadrat. And so these data are still kind of raw. So this would be the number of cover crops of each of those individual cover crops in that quadrat, minus the annual ryegrass, because somehow we missed the counting of the annual ryegrass that was in there. So we counted the cover crops, and ragweed was the main weed present there. So on average, I think there was like one less little ragweed in there in the cover crop versus control. Next slide looks at the heights. We measured the cover crop heights while we were there. Or we also measured cover crop heights, and also missed the annual ryegrass, unfortunately. But just looking at some of the broadleaves there, so on average, everything was about a foot tall or so growing under that corn canopy, more or less. Some of the ragweed escapes were also up there a little bit too. Going on to next slide. So that's some of the, like Michaela had mentioned, the main goal behind this is to look at the soil, how the soil changed from the beginning to the end. We don't really have that capstone and soil data. We won't have that until spring when we do the final soil sampling and get the final soil health samples too. So this is our kind of tentative summary from this year. Like we mentioned, the sunflower impacting the corn, and then I think I forgot to put the total biomass, but it was about a ton per acre of cover crop biomass that we got growing underneath the corn. And would that normally be grazed for you guys? So it would be about a ton per acre of cover crop that would have been grazed with cattle, at least for their operation. So we'll finish up this project next year in the spring, and we will have more to share with you guys then. All right, it is 9:45. So next slide. In the future, we'll be continuing working with that Johnson Sioux bioreactor. We still have at least one more year on that grant. Looking at green seeding soybeans into rye, which you guys will hear more about. Continuing on with volunteer wheat cover crop. We had one site this year. We'll have one site next year. Other things we can look at or continue looking at, if you guys are interested, pivot bio, foliar copper, continuing with the nitrogen rates and the protein mapping. So if you guys like what you're seeing, please write it down. If you have other ideas, please write those down also. If you have any concerns, research you guys think needs you guys have for your farm, please write those down so that we'll bring it up in discussion kind of before lunch here, but that'll help us plan for future years also.